Hello, welcome to the class. Good evening. Am I audible? Boards are clear.
Yes, sir. Good evening.
So we are in class number 2 of Unit 2, core paper. Unit 2, class number 2. And Unit 2 is all about biological classification. In class number 1, biological classification. We classified microorganisms or biological organisms, living organisms into kingdom system. Yes or no? There are various kingdom model, two kingdom, three kingdom. Progressively, it improved into five kingdom model. Yes or no? Which scientist or who discovered the five kingdom model? R.H. Whittaker, Robert
Whittaker.
Whittaker. Okay. And next, the kingdom model is changed into or convert into domain system, right? Domain system, and there are three domains. There are three domains. One is domain eubacteria. Second domain is domain archaea and or archaebacteria. And third domain is domain eukarya, where all eukaryotic kingdoms are put into a single domain and form domain eukarya. What is the basis of domain model? Why kingdom model is changed into a domain model? Answer. Which is best, domain model or kingdom system? Domain,
Sir,
right?
domain system.
Okay, why? Okay, yes. Domain system is totally based on molecular aspect because domain system is created by Carl Woos. Yes or no? Carl Woos, and he created this domain model based on ribosome structure. Based on ribosome structure, and basically in deep into that ribosome has two subunit, small and large, and based on the small subunit rRNA structure. Based on small subunit rRNA structure, these domain models are created, right? And we said that in small RNA, small subunit, the RNA contain either 16S or 18S rRNA. 18S rRNA totally followed by domain eukarya, and 16S rRNA followed by eubacteria and archaebacteria. These are bacterial group. Actually, Monera is split into the archaebacteria and eubacteria by the Calus based on the 16S rRNA nucleotide sequence. Yes or no? Correct. So we are into the domain model, domain comparison model, domain eubacteria, domain archaebacteria, and domain eukarya, and total 11 comparison characteristics we are drawn. Total 11, and this list is not completed. This list is not completed. We are going to complete today's class is totally complete this comparison table because it needs time. The table contain the character. You should know first the character, what is the character means, right? If you say the histone, what is histone, why histones are present, why histones are not present into the separate domain, just needs a simple way the characteristics meaning. That's why it takes time, and we have thoroughly discussed each of the characters, 11 characters. Today we complete this list. Okay, so we are into three domain model comparison, features or characteristics, domain eubacteria, domain Archibacteria, and domain Eukaryote. And we covered total 11 characters. Just recap these 11 characters, the characteristics name. Then we move forward with the character number 12. Character number one was cell structure, right? We compare this character cell structure in between domain Archibacteria, domain Eubacteria, and domain Eukarya. Then we covered the comparison comparison character membrane enclosed nucleus. Membrane enclosed nucleus. Number three, internal membranous organelle. Internal membranous organelle. Number four, its cell wall. We compare between the three domains based on cell wall structure, cell wall composition. Number five, cell membrane lipid. Membrane lipid. All are discussed thoroughly. If you are new to the class, check my video, and its video is uploaded into the YouTube. Membrane lipid. Number six, gas vacuole. Number seven, chromosome structure. Chromosome structure. Number eight, histone protein. Histone protein. Number nine, introns. Introns. And here we discuss a lot about the introns. What is intron? What is exon? Why it is present? Why, etc. All are details we are discussing in previous class. Introns. Number 10, the three process: splicing, capping, and polyadenylation. Polyadenylation. And number 11, mRNA structure. mRNA structure. Right? So these total 11 points, characteristics points that we discussed previous class. And today we start with the new character number 12. Okay? Just recap this point. Character number 12. Character number 12 is tRNA. Is about tRNA. What is T symbolized in? Transfer RNA. Correct. M for messenger RNA, R for ribosomal RNA, and T for transfer RNA. Transfer RNA. Do you know the basic structure of tRNA? Based on class 11-12 knowledge, do you know the basic structure? What is the structure in the tRNA? How does the tRNA structure looks like? Anyone? From online, from offline? Monomoy? Jano. Bullet. You cannot remember this,
Sir,
but you
clover leaf
know.
structure. It was a
Correct,
clover leaf
correct,
structure.
correct, correct, correct. Exactly. The tRNA structure is clover leaf structure. Clover leaf structure that contain four arms. The basic structure is looks like that. Yes or no? This is the tRNA molecule. This is the tRNA molecule where it is a 5' end and it is a 3' end and it looks like a clover shape, clover leaf shape. That's why the structure is called, this structure is called clover leaf structure. And this clover leaf structure has four arms. four arms, right, left, up, sorry, down and up. Clear? So this is the basic structure. Right arm is called T shy C arm. Left arm is called D arm or D loop. And the bottom arm or down arm is called anticodon arm. Anticodon arm. It is specifically for recognizing the codon. Codon, where the codon is lies. Codon lies in the mRNA. That mRNA codon is recognized by this anticodon arm. And the top structure is called amino acyl arm. Amino acyl arm. Because it carry the amino acid. It carry the amino acid. Which specific? This codon specific amino acid carried by the tRNA. That's why it's called transfer. Why it is called transfer RNA? Because it carry or transfer the amino acid molecule from the amino acid full of the cell to the translation site, that is the ribosome. The tRNA function is that to transfer the amino acid to the translation site, which is the ribosome. That is why tRNA is called the symbolized is T with small t is a transfer. Okay, and this is the basic structure, and now we look for the comparison. tRNA comparison between the three domain of life. tRNA comparison between the three domain of life. Okay, first comparison point is thymine presence. Thymine presence. First comparison is thymine, presence of thymine. You clearly see that the right side arm is the T shy C arm where the three nucleotides present T, shy and C. T means thymine. T means thymine. The normal thymine that is present inside the DNA. A bonded with T and G triple bond we see in the case of DNA, yes or no? A means adenine, T means thymine, G means guanine, C means cytosine. So this thymine is present inside the T-shy arm. Normally, thymine is absent in RNA. Yes or no? Normally, thymine is absent in RNA, and in case thymine, in replace of thymine, it present uracil.
Uracil.
But in case of tRNA, thymine, the unusual base here, thymine is present. Look that. Look at that. Thymine is present inside the tRNA. Normally, RNA do not contain thymine, but tRNA contain thymine. tRNA structure has thymine in the T-cell. So presence of thymine, huh? মানে আরএনএ ক্ষেত্রেও আমরা তো সাধারণত জানি আরএনএ তে থাইমিনের পরিবর্তে ইউরাসিল থাকে but tRNA contain thymine. That is the unusuality about the thymine. And you will also study in the molecular biology what is the unusuality about the tRNA. And tRNA one first unusuality is multiple unusual base present. One is thymine. No RNA contain thymine except tRNA. mRNA do not contain thymine. rRNA do not contain thymine, but tRNA in T-cell contain thymine. So domain U bacteria contain thymine, presence or absent? Presence, present. And clearly we see that T-cell arm contain the unusual base thymine. Clear? Whereas Archaebacteria, the unusuality is no thymine is present. The T-cell arm, absence of thymine. Absent of thymine. But eukarya, still thymine present. Check the comparison table. Between the three domains. Is it clear to all? Understood the point? Thymine presence or absent, on that basis, the three domain of life differentiate comparison.
Sir, why in archaebacteria thymine is absent?
That is the unusual. The structure is different from other two life. That's the unusuality of the archaebacteria.
Okay, sir.
Under tRNA, some other comparison character. Amino acid carried by initiator tRNA. Amino acid carried by initiator tRNA. We mentioned that the tRNA molecule is a transfer molecule and it transfers the amino acid from the cell pool to the translation site. So it always carries the amino acid, but the first amino acid is denoted for the translation is always methionine. In some case it is methionine. In case of bacteria it is formal methionine. So compare is between methionine is the first amino acid or formal methionine. Formal methionine. So the amino acid that is carried by the first tRNA molecule, initiator tRNA, the first tRNA required in the translation is called the initiator tRNA. So initiator tRNA carried the first amino acid, either methionine or formal methionine. Between that, the comparison is given. The domain eubacteria always carry formal methionine. Formal methionine. And the three-letter code of methionine is mate. You can also write formal mate. It is absolutely correct. It is universally accepted three-letter code. Short form of methionine. It's accepted. You can write. হ্যাঁ না ওটা হবে না ওটা ফুল ফর্ম লিখতে হবে Archaebacteria, it is methionine. It is methionine. Not formal group is added. Archaebacteria carry actual the methionine. No formal groups is added in case of Archaebacteria. But Eukarya, it is also methionine. It is also methionine. Only in case of eubacteria, the first amino acid that is attached to the protein is N-formal methionine. So character number 12 is the tRNA, and out of tRNA we compare two sub-character. One is thymine is present or not, and where the thymine is present, you should for that you should know the basic structure, and we clearly see that the T caesium contain the thymine. Eubacteria, Eukarya contain thymine in T caesium, but Archaebacteria is unusual. It do not contain thymine in T caesium. First, and difference second number is what amino acid is carried out by the first tRNA. That's called initiator tRNA. The initiator tRNA means the first tRNA that is involved in the protein synthesis. That is the initiator tRNA. The first amino acid is, in case of eubacteria, it is different. It is different methionine, but formal group is attached. Formal group is attached to the methionine, and that is the first amino acid in case of eubacteria. Archaebacteria and Eukarya, it is methionine. Clear? Point clear? Online students, point clear? Everybody?
Yes, sir. Yes, sir.
Okay. Next character is the ribosome, and there are also multiple sub points. Okay. Number 13, comparison list. Ribosomes. First of all, size. First of all, size. The size of the eubacterial ribosome. The size of the eubacterial ribosome is, you all know that. Mention.
Yes.
Small. Total subunit. Total subunit size. Total subunit size. Online students, total subunit size of the ribosome in bacteria.
30 is the 55 is.
50s, small and large, and combined together to form 70s. 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 The size of the ribosome in eubacteria is 70s. Archaebacteria, always size is same, but structurally it could be different. So structure size is almost same with the bacteria. Eukarya, it is
It is.
80s. The size of the ribosome is 70s, 70s, and 80s between the three domain of life. Second one, second subcharacter, subunits. Subunits. Domain eubacteria contains small subunit and large subunit. The subunit size is 30s and 50s. Large 50s, small 30s. Correct? Archaebacteria, same size. Large 50s, small 30s. Eukarya, the total size is different. That's why the large and small subunit also be different. Large subunit and small subunit, one is 60s, other is 40s. Large subunit is 60s and small subunit is 40s. Size, just size. Clear? Structural composition. Ribosome structural composition. How does the ribosome is made? Ribosome is totally made up of riboprotein, ribonucleoprotein. Ribosome is made up of ribonucleoprotein. Made up of ribonucleoprotein. That means if you break down the word, you get two substances. One is ribonucleo, other is protein. So ribonucleo means RNA, because RNA full form ribonucleic acid. So that frame, word ribonucleo means it's connected to the RNA, and another substance is clearly it is mentioned here. It is protein. So two structures composition contain, ribosome composition contain two structural part. One is RNA, other is protein. And now question is which RNA? Which RNA? rRNA, tRNA or mRNA? Clearly we see that tRNA has different function. mRNA has different function. So which RNA present inside the ribosome? It is rRNA. It is rRNA, and R designate as ribosomal. R designate as ribosomal. For that it is named as rRNA. So structural composition contain rRNA and protein. rRNA and protein. Clear? Now compare. Compare rRNA between the three domain of life and protein in three domain of life. Okay. rRNA, the large subunit of the eubacterial rRNA, large subunit, that is the 50S. 50S contain 23S rRNA and 5S rRNA. Clear? The large subunit 50S contain 23S rRNA and the 5S rRNA. And the small one, in the small subunit 30S contains 16S rRNA. Look at that. These 16S rRNA actually are sequenced by the Carl for their 3 domain. This 16S rRNA. Where it is present? It is present in the small subunit of the ribosome. Clear? This is the rRNA of eubacteria. Archaebacteria. Archaebacteria. The large subunit, which is 50S, contain 23S, correct? 5S, as well as some of the archaea contain additional rRNA, which is not found in the bacteria. Additional rRNA called 5.8S. 5.8S rRNA. Some archaebacteria also contain 5.8S rRNA. Look at the difference. Eubacterial large subunit contain 23S and 5S always. You can check any bacteria and you find that it is contain only 2 rRNA, 23S and 5S. But Archaebacteria has the problematic. Not problematic means changes. Many archaea contain extra rRNA, which is 5.8S. Clear? And the small one contains similar, only single rRNA, that is 16S rRNA. That is 16S rRNA. So there is no difference between the small subunit, but there is difference in large subunit rRNA composition. Clear? Eukarya. Eukarya large subunit contain 28S rRNA, 5S rRNA and 5.8S rRNA. 28S, 5S and 5.8S. And you can see that this 5.8S also found in Archaebacteria. This 5.8S also found in Eukarya also. Yes or no? Look at the 5.8S. And clearly absent in 5.8S in Eubacteria. Okay. Small subunit contain 18S rRNA. Contain 18S rRNA. So rRNA composition based on comparison between the rRNA, 3 domain of life. Clear? Here also the similarity. Mark the similarity between Archaebacteria and Eukarya. Clear? Protein. Protein. The ribosomal protein contained in the eubacteria, total ribosomal protein is 55. Approximately total 55 R protein. R protein means ribosomal protein. 55 ribosomal protein approximately found in eubacteria. But Archaebacteria, though it is similar size, but the protein content is almost greater than the eubacteria. Eubacteria contain 55, but Archaebacteria do not contain it more than the 55, and it almost 68. 68 proteins found in Archaebacteria. Though the size is similar, look at the difference. It can be size or it can be similar, but the internal composition has changed between bacteria and archaebacteria. And eukarya, the size is greater, so almost protein is higher side. That means total 80 protein, approximately 80 ribosomal protein found in eukarya. 80s RRN. 80s ribosome. Comparison point clear. Structural composition. Various type of question can be come from that chart. It's a very important chart. Online students understood, points are understood.
Yes, sir.
Okay. Ribosome point subpoint 3. Subpoint number 3. Diphtheria toxin reactivity. Diphtheria toxin reactivity. Diphtheria toxin actually coming from the bacteria Corynebacterium and diphtheria. Corynebacterium and diphtheria produce diphtheria toxin and cause the disease. How? This toxin bind to the ribosome and almost stop the translation process. Ribosome is the hub of the translation, site of the translation, that means protein synthesis. So diphtheria toxin when present, it bind to the ribosome and completely shut down the translation process, the protein synthesis process. By that way, cell is killed and tissue organs simultaneously become killed. That is the diphtheria disease progression. Okay, so how does the diphtheria toxin work? Diphtheria toxin works by binding to the ribosome. Clear? Binding to the ribosome. Which ribosome? Prokaryotic or eukaryotic ribosome? Prokaryotic the bacteria kill হয়. If it work on the prokaryotic ribosome, then the bacteria will be killed. So it bind to the eukaryotic ribosome. Diphtheria কাদের হচ্ছে? Human and other animals. That means eukaryotic animals, eukaryotic organisms. So diphtheria toxin, where does it bind? It bind to the eukaryotic ribosome. Clear? Online students clear? And by that way, this toxin created the problem. Okay? So domain eubacteria, reactivity occur or not? No clear answer, because it only react with the eukaryotic. Answer is no. No reaction, no reactivity. Yes or no? ठीक?
Yes.
Correct. Answer. Online students. Correct. No reactivity inside the in case of eubacteria.
Yes, sir.
Archaebacteria. Interestingly, the activity found. Interestingly, the activity or reactivity found. Yes. No suicidal type, because diphtheria toxin is created by the Corynebacterium and diphtheria. It is under the eubacteria, not the Archaebacteria. So it is not suicidal. The domain is different. Corynebacterium and diphtheria is the eubacterial organism. So interestingly, diphtheria toxin, the activity occur with Archaebacteria. Though the ribosome size is same, but the internal structure has different. That is why diphtheria toxin react with Archaebacteria, not eubacteria. Clear? In case of eukarya, obviously reacted. Point clear? Comparison point clear?
Yes, sir.
Mark those points. Where Archaebacteria and eukarya shows similarity. Mark those points. কেন মার্কিং করতে বলছি একটু পরে বুঝতে পারবে, যে কোশ্চেনটা কেমন ভাবে ঘোরাবে চেক করবে। At the end we are saying the question or type of question where it needs the similarity between archibacteria and eukaryote. Okay. So diphtheria toxin reactivity done. Next, under ribosome subpoint, chloramphenicol kanamycin reactivity. Chloramphenicol reactivity. Chloramphenicol is the antibiotic, is a bactericidal. That means chloramphenicol is the antibiotic and bactericidal.
Bacteria also work.
Bactericidal. Cidal means killing activity. Killing activity. Which organism it kill? Clearly mentioned in the prefix bacteria. That means bactericidal. So chloramphenicol reacted with which domain?
You bacteria.
Eubacteria clearly yes or no?
Yes.
Here it is yes or no. Chloramphenicol reactivity you found in the eubacteria or not?
Yes.
Yes, because chloramphenicol always react with the bacteria and try to kill them. So yes, correct. Archibacteria, interestingly, it is no. It is no. So chloramphenicol cannot work in archibacteria. Eukarya, obviously no. Obviously no. Yes, chloramphenicol bind with the ribosome structure. Chloramphenicol, the antibiotic, this antibiotic, how does it kill? It bind to the ribosome of the bacterial ribosome. Bacterial ribosome and try to eliminate the function of ribosome. By that way, protein synthesis has been stopped in case of bacteria. And you will see that the archibacteria also do not react. Only eubacteria is reactive. So what is the basis? Because of the changes in the protein. Changes in the protein, changes in the 5.8S, mainly protein. Why chloramphenicol reactivity? Another potential question. Why chloramphenicol reactivity not found in the archibacteria, though the ribosome has the similarity in the size. Both are 70S, both are 70S structure, size, but chloramphenicol activity, the activity is not found in the archibacteria. Why? Because of different protein structure. Protein structure has been more and more in case of archibacteria. Almost 68. 68 types of proteins are present. Clear? So protein significantly differ these characteristics. This is also same. Diphtheria, why diphtheria toxin reacted in archibacteria? Because of the protein. Clear? Point clear? Next character, kanamycin reactivity. Another antibiotic, another bactericidal. So state the comparison. State the comparison. In first column, yes or no?
Yes.
In first column, yes or no? Yes. Reacted. Bactericidal. Archibacteria, surprisingly no. And eukarya, obviously it is no. Clear? Another similarity. Online students, can you get my point? Can you get my point, online students?
Yes, sir.
Answer, or state something. Next, next point. Another drug. Amizomycin. Amizomycin reacted with eukaryotic ribosome. Reacted with eukaryotic ribosome. So it is a drug for eukaryotic cell. So now state the comparison.
No, yes, yes.
No, surprisingly it shows yes, and obviously it is a eukaryotic drug, so it binds with the eukaryotic reaction. Clear? Ribosome points are done. Point number 13, ribosome complete. Now we can relate to point number 14. Point number 14, DNA polymerase. Point number 14, DNA polymerase. Do you hear this enzyme previously? DNA polymerase. What is the function of DNA polymerase?
Binding.
Where
Um, yeah, like you. No.
Sir, in transcription.
transcription RNA polymerase. DNA polymerase is
Sir,
in
replication. Sir, replication.
replication. Doubling of DNA from one DNA strand to another DNA strand is the semi-conservative way to convert into another, and that time the enzyme that is needed. One of the enzyme. There are various lists of enzyme. One of the main enzyme is the DNA polymerase. Here is the term DNA. That means it work on DNA to make another molecule of DNA. That's why it is called DNA polymerase. And polymerase means polymerization word, nucleotide addition word. Okay, so DNA polymerase, the main DNA polymerase in domain eubacteria is polymerase 3. Pol 3 is stated that Pol 3. Various type of DNA polymerase is present, but the main polymerase in case of domain eubacteria is Pol 3. Okay, multiple polymerase are there, Pol 1, Pol 2, Pol 3, Pol 5, Pol 4, but the main, here is the main DNA polymerase. Yes, write down the characteristics here. Main. So main DNA polymerase in case of eubacteria is Pol 3. Main DNA polymerase in case of archibacteria, it is Pol B. Pol B. And in case of eukarya, it is Pol alpha. Pol alpha and Pol delta. Pol alpha and Pol delta, because Pol alpha work on lagging strand. Work on lagging strand. And delta work on leading. Lagging strand synthesis. And Pol delta leading strand synthesis. What is lagging strand? What is leading strand? In DNA replication, based on class 11-12 knowledge, you should answer what is lagging strand, what is leading strand. কি হবে? স্যার বায়োলজি পড়েছি, বাট ওভাবেই পড়েছি শুধুমাত্র আনসার করার জন্য, দেখতে ঢুকিনি তো, আর বোঝারও সেরকম চেষ্টাই করিনি। স্যার বলে গেছে, স্যার ম্যাডাম স্কুলে বলে গেছে, টিউশনে বলে গেছে, আচ্ছা ল্যাগিং লিডিং হয়, কি ব্যাপার? ঠিক আছে, চলো ওটাই দেখবো, মানে জাস্ট লিখে নেবো। ল্যাগিং অ্যান্ড লিডিং, লিডিং মানে লিড করছে, লিডিং, দ্যাট মিনস ইট প্রোগ্রেস fast. The strand which is progress fast, that is called leading strand, and it is the strand of 5 prime to 3 prime. Okay, that is another details. Lagging strand is progress slowly, lag behind. That means slowly progress করে, and it progress in a fragment manner, and that fragment is called Okazaki fragment. Okazaki fragment found in lagging strand. Because it lagged behind, it slowly progressed and in progress like a fragments, 1000 to 2000 nucleotide fragments. So this lagging strand and leading strand have for the synthesis in eukarya two different polymerases. But in case of archaebacteria and eubacteria, you will see that this leading and lagging strand both are strand copied by single enzyme. Polymerase 3 here and archaebacteria Pol B. But in case of eukarya, it needs two different enzyme for synthesis. Clear? Point clear? Online students, points clear? DNA polymerase point,
Yes, sir.
family of DNA polymerase. Family of DNA pol. Various families are there. The main enzyme fall in eubacteria family C, C family, capital C. The main enzyme in case of archaebacteria fall in B family. That's why it's called B, Pol B. So it is in from B family group. Clear? And eukarya, the main enzyme fall in also B family. Also B family. Another similarity. Another similarity. The main enzyme, the main enzyme in case of archaebacteria and eukarya is coming from B family, category B family. But eubacteria is from, main enzyme is from C family. Point clear? Next point, point comparison number 16. RNA polymerase. RNA polymerase. RNA polymerase. What is the function of RNA polymerase? What did he do? What did it do
RNA replication.
to make
RNA.
RNA from DNA?
Yeah.
From DNA, it make RNA from DNA. So there are some points. Subpoint number one, number. How many number are RNA polymerase found inside the three domain of life? And it is clearly a single RNA polymerase that do all the work in case of eubacteria. Just only single one RNA polymerase, that is the hero enzyme. It can do, it can form synthesize rRNA, it can form mRNA and tRNA also. So single RNA polymerase can do all the work. It's a single one. Clear? But archaebacteria, it is also single. It is also here also a single enzyme can do all the work. But eukarya needs more than one, three enzymes, three RNA polymerase. RNA pol 1, pol 2, and pol 3. RNA pol 1, RNA pol 2, and RNA pol 3. Clear? Second sub-character, rifampicin, another drug. Rifampicin, sensitivity. Rifampicin, another antibiotic. It works on prokaryotic RNA polymerase. Which RNA polymerase? It works on prokaryotic, means bacterial RNA polymerase. So now state the comparison table. Where is the sensitivity lies?
Actually, RNA.
Bacterial RNA polymerase.
Roman evacuate yes or no?
Yes, our sensitivity yes, because it react with the bacterial RNA polymerase and stop the reactivity, stop the function. Yes, Archibacteria. Surprisingly,
No.
no. Surprisingly, no. And Eukarya,
No. Yes.
obviously no. Clear? So from that view, we can also mean it means that the RNA polymerase, though it present in single from Archibacteria and Eubacteria, but structurally it is different. That is why rifampicin could not work on Archibacterial RNA polymerase, but it can work on Eubacterial RNA polymerase. So structural difference is lies in between Eubacteria, Archibacteria, RNA polymerase. Yes or no? The statement is correct or not?
Right.
If the statement is given, RNA polymerase structure of Archibacteria and Eubacteria is different. Most of the students are giving answer no, as it mentioned is single one, but no, it is wrong answer. Different lies. Eubacteria, Archibacteria, different lies. That is why rifampicin could not work. Clear or not? Understood my point? Online students all are muted. Points clear?
Has sir, clear.
Yes, sir. Yes, sir.
Okay. RNA polymer is done. Next point. Point number 70. RNA polymerase 2 type promoter. 2 type promoter. This promoter is not that produce a building. This promoter means it is the starting point of the RNA synthesis. The starting point of the RNA synthesis, and the starting point is different in based on the RNA polymerase. RNA polymerase 1 has different promoter, 2 has different promoter, 3 has different promoter. Clear? But as Eubacteria, Archibacteria contains single promoter, single RNA polymerase, so the promoter should be one, just one type. So there is the point. RNA polymerase 2 type promoter. Obviously, Eubacteria contain or not?
Yes.
No, because it contain only one type promoter, as it is single. So polymerase 2 type promoter not found in the Eubacteria. Correct? And as well as Archibacteria, it should be no, but surprisingly,
Yes.
Pol2 promoter found in some Archibacteria. Yes, clear? And Eukarya, obviously, answer is yes, Pol2 promoter, because there is a RNA polymerase 2. There is RNA polymerase 2. And here is only Pol1, polymerase 1. Single type. So only one type of promoter is found. But surprisingly, in case of archibacteria, Pol2 promoter also found. And it is another similarity between archibacteria and eukarya. Rifampicin sensitivity also similarity point. Mark. Done. Any question? Any question? Online
No, sir.
students. Point number 18. Metabolism. And there are also several subpoints. ATP synthase. Did you hear this enzyme before? ATP synthase,
We had ATP synthesis.
ATP synthesis, FOF1 ATP synthase. The full form is FOF1 ATP synthase and make the ATP inside the
तो mitochondria देखो कहीं
mitochondria as well as in bacteria also ATP synthase found that produce ATP. So bacteria needs ATP, na? So bacteria also found ATP synthase, but the structure there is a difference or not. That is the comparison part. So ATP synthase, eubacteria, the structure is different. Archaebacteria, similar. Eukarya, similar. Another interesting similar characteristics. Similar characteristic. ATP synthase structure. Methanogenesis. Methanogenesis. What is methanogenesis? Is the methane production capacity. Methane production CH4. Methane production capacity. Methanogenesis cannot produced by any eubacteria. So no. Archaebacteria, yes. This is the bacteria that produce methane. And you also see that the methane is produced inside the rumen system. That means the cow produce methane inside the rumen, inside the stomach. Yes or no? So how does the cow produce methane? Because of the rumen contain archaebacteria. So cow does not produce methane. Actually, methane is produced by bacteria remain in the rumen system, remain in the intestine of the cow. Clear? So these bacteria, these archaebacteria produce methane and it is involved in the biogas production also. This methane is involved in biogas production. Okay. Eukarya. No, no, no. No eukarya cannot produce methane. So methanogenesis activity do not found in eukarya, eubacteria, but only found in archaebacteria. Nitrogen fixation capacity. Nitrogen fixation capacity. Eubacteria, yes or no? Bacteria has the nitrogen fixation capacity or not? Yes, yes. Rhizobium, nitrogen fixation capacity. Azotobacter, nitrogen fixation capacity. Cyanobacteria contain nitrogen fixation capacity. So eubacteria, yes. And some archaebacteria also has nitrogen fixation capacity, but no eukarya, no eukaryotic organism has the capacity of nitrogen fixation. That is why plant need nitrogen fixing organism for their better fertility. Better soil fertility needs nitrogen fixation capacity organism. Clear? Can you answer why eukarya cannot do nitrogen fixation? Anyone guess?
Even bullet.
Maybe.
Plastic.
Maybe. Yes. 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 This nitrogen has 3 bonds. N triple bond N is a very strong bond and cannot be broken down by any eukaryotic organism. That is why the prokaryotic organism is the capacity has the capacity to convert this nitrogenous compound into atmosphere into different product, reduce the product into NH4, NH3, but it cannot be cleaved by or converted by eukarya. Next, chlorophyll based photosynthesis. Now compare with that character.
Bacteria তো করতে পারবে।
করতে পারবে না? Yes, chlorophyll present in some bacteria. Yes, chlorophyll B, chlorophyll A found in bacteria. Some bacteria has the capacity of photosynthesis. So these bacteria contain chlorophyll. In fact, cyanobacteria contain chlorophyll. Plant like chlorophyll found. Eukarya, obviously yes, because all plants contain chlorophyll. And fungi, algae contains chlorophyll. Some protista also chlorophyll containing. Archaebacteria cannot. No archaebacteria has chlorophyll. Next sub point, chemolithotrophy. Chemolithotrophy. Popular term, chemolithotrophy in unit 1, and these organisms are described by which scientist? Chemolithotrophy. Now the compare point.
Bacteria part.
Eubacteria parve. Archaebacteria also yes. Some archaebacteria has the capacity of chemolithotrophy, but eukarya yes cannot. Clear? Next and last character point, number 19, penicillin sensitivity. Penicillin sensitivity. What can be the compared in eubacteria, archaebacteria, and eukarya? Penicillin sensitivity, yes or no? Eubacteria, no. Eubacteria, yes. Penicillin works on bacteria. Penicillin, yes, works on bacteria and kill the bacteria. In which part penicillin works? It's in the cell wall. Works on cell wall of bacteria. Clear? So penicillin sensitivity present in eubacteria, yes or no?
Yes.
Yes. Archaebacteria cell wall structure is similar or different?
Different.
Cell wall structure is different. So penicillin is working on archaebacteria or not?
No.
No.
But
No, because structure is different.
yes.
It's a pseudomurin structure. Archaebacteria cell wall composition is pseudomurin, and eubacteria is a not normal peptidoglycan or murin. So the structural changes and penicillin could not work on archaebacteria. Whereas eukarya, obviously no, penicillin do not work on eukarya, only work on eubacteria. So another interesting similarity between archaebacteria and eukarya. Though they contain peptidoglycan, but peptidoglycan is a pseudopeptidoglycan and could not react with penicillin, which react on eubacteria easily, but archaebacteria do not react.
तो केमिस्ट्री में ज्यादा एक्सेप्शन हो गया ना केमिस्ट्री
Exception, that's the interesting, surprising difference, contrast. All points are clear? Online students, all points are clear?
Yes, sir.
Now try to answer two important question. দেখো কোশ্চেন কিভাবে ঘোরায় ক্যালকাটা ইউনিভার্সিটি কোশ্চেন কিভাবে চেঞ্জ করে নরমাল কন্ট্রাস্টিং পড়লে তো কম্পারিজন পড়লে বা কোশ্চেন চেক করো তোমার মাথাটা কিভাবে খাবে দেখো Question number 1. Compare between three domains of life based on central dogma. Completely twist করে দিল দেখো
যে মানে সেন্টার ডগমা মানে যদি কেউ না ধরতে পারে তাহলে
শেষ শেষ done. Based on central dogma, that word completely changes the whole scenario of the question. Yes or no? First when you hear, oh, it's a easy question, compare three domains of life, but tricky gives the word central dogma. So what central dogma means?
replication
Central dogma means replication,
translation
transcription, translation. Correct?
yes
That means
Yes.
simply we can say that central dogma means DNA to RNA conversion, RNA to protein conversion. Correct? And DNA to DNA conversion. So DNA to DNA conversion occur by replication. DNA to RNA conversion occur by transcription, and RNA to protein conversion occur by translation. So you should write those points that is based on central dogma. That means transcription, translation, replication, DNA, RNA, chromosome structure, RNA structure, protein structure. আচ্ছা আমার লিস্টের মধ্যে ছিল all points are given but you should pick those point only. So কি পয়েন্ট আমরা নেব ডিএনএ স্ট্রাকচার আমরা নিতে পারি মানে ক্রোমোজোম স্ট্রাকচার পয়েন্ট আমরা নিতে পারি yes or no?
yes
We can take chromosome structure point. Clear? We can take RNA structure, rRNA, tRNA, mRNA. প্রোটিনের সেরকম কিছু স্ট্রাকচার আমরা চেক করাইনি বাট রেপ্লিকেশনে ডিএনএ পলিমারেজ we can take DNA polymerase point. Transcription we can take RNA polymerase point yes or no?
yes
Yes, sir.
Translation we can take ribosome point, various ribosome point, subpoints, antibiotic sensitivity. তো তুমি যদি অন্ধের মতো প্রথম থেকে শেষ অবধি লিখে যাও
নাম্বার
সিইউ বসে আছে কাটার জন্য টিচার্স আর ওয়েটিং ফর ইওর আনসার। তুমি যদি এখানে সেল ওয়ালে কম্পেয়ার করো, সেল মেমব্রেনে কম্পেয়ার করবে, একটা নাম্বারও দেবেন, বিকজ ইটস টোটালি বেসড অন you should write the compare list based on central dogma. So what is central dogma? Central dogma means you can pick DNA structural point, RNA structural point, replication point, transcription point, translation point.
এখন শুধু সিও না, সবাই মানে কাউন্সিল এবারে করেছে। আপনি আশা করে দেখলেন 2024 9 মাসের একটা সেকেন্ড কোশ্চেনটা আপনারা যদি দেখেন, ওরা কিন্তু ডাইরেক্ট আমরা বিগত বছরের ট্রেন দেখেছি, এবারে ওরা ভিতরে পয়েন্ট তুলে কোশ্চেন দিয়ে দিয়েছিল।
আর সিইউ তো আরো বেশি করবে, তুমি একটা এক্সাম দাও তাহলে বুঝবে। সিইউ ইজ আ ট্রিক করবেই কোশ্চেনে, ঠিক আছে, স্ট্রেট ফরওয়ার্ড কম দেবে কোশ্চেন। এন্ড এই কোশ্চেনের প্যাটার্ন বুঝতে গেলে তোমাকে ইনডেপথ স্টাডি করতে হবে। ওভার স্টাডি করে যাবে তো আনসার করা ডিফিকাল্ট হয়ে যাবে। দেখো তুমি পড়ে গেছো, থ্রি ডোমেইনের কম্পারিজন পুরো লিস্ট নিয়ে চলে গেছো, ধরো তোমার কাছে পুরো লিস্ট আছে, বাট ইউ ক্যান নট আনসার ইফ ইউ ডু নট নো দ্য টার্ম সেন্ট্রাল ডগমা। তোমার কাছে আমি যদি লিস্টও দিয়ে দিই, পুরো 1 পেজের লিস্টটা, যেটা 22 ইয়ে, তুমি আনসার করতে পারবে না। হ্যাঁ কি না? ইয়েস অর নো? সো দ্যাটস দ্য ইস্যু। ওকে, কোশ্চেন এটা হোমওয়ার্কে রাখবো। Question number 2. Archaea is more phylogenetically closer to eukarya than bacteria. দেখো কোশ্চেন কিভাবে চেঞ্জ করেছে, এক্সপ্লেইন। তোমার কাছে কম্পারিজন লিস্ট থাকলেও তুমি আনসার করতে পারবে না। Archaea is more phylogenetically closer to eukarya than bacteria. And from our comparison list, it is very clear. It is very clear that many of the structural aspect, molecular biology structural aspect, the archaea and the eukarya similar. Yes or no? So we can safely say that actually archaea produces eukarya and bacteria is totally separated domain. Totally separated domain. Archaea evolve into eukarya later. So archaea and eukarya are similar. Closer, not similar, closer. Phylogenetically more closer. Ancestrally more closer than bacteria, because bacteria is totally different points. All points are those similarity in case of archaea and the eukarya, these points are totally different from bacteria. So what are the points you pick for to answer this question? Those is the marking point where similarity lies between eukarya and archaeobacteria. That means sensitivity, drug sensitivity points, tRNA structural point, etc., etc. Those are similarity points. So you should pick similarity points between archaea and eukarya. And there are almost 8 to 10 similar points is in the list. These two questions are in the homework. Previous year question. See. তোমার কাছে রেডি থাকলেও তুমি আনসার করতে পারবে না if you do not know or do not think about the question, what question wants. উল্টো পাল্টা লিখে আছে নাম্বার দেবে না। ক্লিয়ার? পয়েন্টস ক্লিয়ার? Domain concept clear? Domain comparison points clear?
Yes, sir.
Okay. So next day we cover about some details of archibacteria. So next day topic is about archibacteria.
Sir, I
Variety of
have a
comparison
question.
person.
Sir, is there any test on Friday for the sec paper unit 2?
Friday, not Saturday. No, as the notes are still pending, so that is why we cancel this Saturday exam. After notes given, I will give you 1 week. Then the exam is conducted. Schedule.
Okay, sir.
Clear? So archibacteria, next day topic. Any question, any doubt from this class?
No, sir.